Peculiar thing about the rabbits is that Soviets picked rabbits strictly for practical purposes. But it turned out that a rabbit is extremely safe animal. Every textbook of veterinary medicine will tell you that there is no viral infection of rabbits which is transmissible to man. Not a single one. There are some bacterial, well-known diseases that can be transmitted, but the rabbit is, so obviously sick that you cannot possibly do that. Number two is, when you are using the rabbit fetuses or even newborns, a rabbit fetus, when you are taking it out, is sterile. Absolutely 100% guarantee, and so is newborn. It takes about 3 days for the newborn rabbit to get colonized by bacteria from outside. But at the moment of birth, it is absolutely the same as fetus. And the last item is, as a mammal, like any other mammal, rabbits should have retroviruses. Okay? Pigs were disqualified as a source for cells, because in pigs they were found 4 different kind of retroviruses that are transmissible to men. So pigs are out of game completely. In rabbits, they should have retroviruses, and they, but everywhere in every publication find no one can find them. It's a question nobody can answer, but as a result of it, rabbits are number 1. The safest animal is out there. Now, you certainly cannot say that about human fetuses, because we know of the cases where simply, a human fetus was used as a source of, different things for the body and transplanted to the patients, or from adult patients, and the patients that were recipients of these different, transplants, or picked up AIDS, because, the patient, in this hurry that takes place, when you get that body, and all of that, were simply insufficiently tested. Okay. Now, with rabbits, it cannot happen. That is the nice thing, because medical legal, if you can imagine, that is tremendous point. That even FDA tried, and they published their own study about the, retroviruses in rabbits, and they have to admit they didn't find them. So it's one of the, in my book, all of the things I mentioned, okay? In nature, it is like that. This is very interesting. That in nature, there is a natural barrier which prevents transmission of infections. You would not recognize difference between domesticated rabbit and a wild hare running around in Australia. You put it on the table, the same animal. If you can imagine, you cannot transmit any infection from domestic rabbit to wild hare, or from wild hare to domestic rabbit. You can put them in one of them can suffer from the deadliest of infections, the hemorrhagic pneumonia, and put him in the cage with the other rabbit, the wild with non-wild, that is perfectly healthy. That first one will die, and the second one will walk away in perfect condition. Kind of, I would say, interesting. The barrier is much more powerful the more distant the animals are. Since rabbit is, shall we say, distant to human being, okay? The chance of actually transmission of any infection from rabbit to human being is minuscule. And likewise, the human being cannot actually infect it very much the rabbit, but fetal and newborn rabbits are very healthy, all right? Now, a very important matter is, as I already told you, that about the different cell types that we have, we have had made. They are making our body and body of mammals. Is sum total of this on cytological level, on the level of cells. That's our baseline. Okay. Every member of animal kingdom is the same. All right. If we are talking to going into lower type animals, various vertebrates, fish, and birds and all of that, they may not need 200 cell types to construct their body. The number is will be lower. But those 120 that they are, necessary to make the body of fish, they're exactly the same 120 that we are using for construction of human being, except that we need additional 120 cell types or something like that. Okay, so that's why you can in reality make cell transplants from any vertebrate. You can make them from fish. There was a study that originates from Chicago by one biochemist, Steiner, who studied insulin in animal kingdom, and he found fish makes insulin too. All right, and the difference between you see, insulin consists of about 188 amino acids. It's a chain. Okay, now the difference between human insulin and rabbit insulin are 2 amino acids. Difference between human and pig insulin, which has been used in medicine, is still being used, 2 amino acids. Difference between human insulin and fish insulin, 2 amino acids. So it's not the issue of in other words, you select you can use rabbits for the same purpose, but the rabbit is too small. How much insulin can you squeeze out of the poor thing, as compared with pig? So as a result of it, this is how it is in nature. And and all of these facts are known, except not passed around sufficiently. So the point is why it was sort of important to reach the point to be able to prove to people that animal, fetal precursor stem cells are the same as human, and so on and safer is because in the end, human fetal cell transplantation has no future, because you cannot get enough fetuses in any country of the world, to actually make this treatment of commercial interest even. See? While with animal, you in reality could set up a semi-industrial system. The one reason it will never go into be complete industrial system is one and only, you see. In clinical side of the cell transplantation, whether human or animal makes no differences. This is and always has been individualized therapy. Okay, you are not giving every diabetic the same combination of cells. You analyze every case, by going through all the 15 systems to pick up all the individual differences from one diabetic to the next. And then you make, and you don't go by diagnosis, you go by pathophysiology. Yeah. You may know that a diabetic has problems in 6 organs. It is the islet of pancreas, where the beta cells are making the insulin. But the alpha cells are making glucagon, another hormone extremely important in metabolism of sugars and lipids. And there is another type of cell in the islets of pancreas that makes somatostatin, again involved in the same metabolism. So from islets of pancreas alone, there are 3 different hormones. The interplay between those is very important. Now, every diabetic will have liver in bad condition, because liver is the chief organ of metabolism, and liver has to compensate for all of this metabolic mess in the body of both carbohydrates and mess. As a result of it, every diabetic will have liver stuffed up with fat. It's called steatosis of liver. As a result of it, the liver will be malfunctioning. And this fat in the cells will trigger scar tissue production around the cells. So 90% of patients are going to fibrosis of the liver. So the liver is actually in bad shape. So when you treat diabetic, you must always treat liver as well. Next problem is adrenal cortex, another endocrine gland. Adrenal cortex is where this cortisol is made, right? Because diabetes is such a constant stress on the body, the adrenal cortex in every diabetic after many years of diabetes will be exhausted. And you will find by analyzing it that the endocrine function of the adrenal cortex will not be normal. So when you are treating the patient, you must give them also stem cell transplants for the adrenal cortex. Next comes our hypothalamus, which you already discussed. All right? Hypothalamus is under tremendous, stress, in diabetes. It has to compensate and compensate and compensate. And, it's like if you're on a crazy square and there is one policeman for all of the tremendous traffic, It's a tremendous amount of work. You must treat the hypothalamus of diabetic if you want to have a successful treatment. Then next item is the diabetes kills the microcirculation. That is the key problem of diabetes and all of the diabetic complications. We know only one cell transplant which will heal microcirculation. Placenta, particularly the cytotrophoblast of placenta. Regardless whether the source of the trouble with microcirculation is diabetes or is something else, okay? Each such patient must receive placenta, okay? And it is a spectacular, result. If the patient receives, that has, for example, bad circulation in his feet, you give patient, this implantation, and then you go to see him in his bed 1 hour later, and his cold feet will be warm already as a first reaction, you see. And the last item that we give to the patient is a lining of the stomach and duodenum, the first part of the intestine. Why? Because, of course, ideally we should give patient stem cells of the islets of pancreas. But stem cells of the islets of pancreas are not in the islets of pancreas. That is definitely known. Nobody knows exactly where are they because nobody has found them yet, but embryologists say that as close as they get to the point, they are in the lining of the lower part of the stomach and in the very first part of the duodenum. Those cells that cannot be separated from the rest, there are among them stem cells. So based on that, we create a preparation from this area and give it to the patient. Right? So this is, I'm giving just one example how the prescription is made. But then the diabetic may have other problems. Many have trouble with the heart muscle. Very common. Many diabetics actually have problems with the brain, and so on and so forth. So you have to examine patient in great detail and give the patient everything the patient needs. Okay. And that is what we call pathophysiological approach to diagnosis. When you do the diagnosis like this, that from the diagnosis you automatically see the prescription. What cell types this patient has to receive. Okay. So there are no fixed prescriptions for anything. That is the key to success.